Welcome, students, to a brief lecture on the histology of the urinary system. Of course, one of the primary things we're going to look at when we are looking at histology and tissues of the urinary system is the kidney. So one of the things that you want to be able to do is to differentiate between the outer cortex of the kidney and that inner medullary layer. So the cortex is where we find all of our nephrons, and so you're going to see all of the renal corpuscles and cross sections of the renal tubules. And then down in the medulla is where we're mostly finding the collecting ducts, and so you'll just see kind of just rays and rays of simple cuboidal epithelium that makes up the walls of the collecting ducts. You do also want to differentiate between the different types of nephrons. So the cortical nephrons, remember, are the ones that sit really high in the cortex with the kind of the whole of the nephron in the cortex. And so if you're looking at a slice of kidney tissue and all you see is corpuscles and tubules, then you're probably looking at a cortical nephron. But if you're looking at a slice of tissue and a slide of kidney tissue and you see both renal tubules and renal corpuscles and collecting ducts, then you're probably looking at one of these juxtamedullary nephrons that are sitting lower in the cortex, juxta next to the medulla, and so you're getting some of that medulla in your slide as well. You also want to be able to differentiate the different parts of the renal corpuscle. So the corpuscle itself consists of the glomerular capillary, which you can see here, as well as the capsule around it, the glomerular capsule or the Bowman's capsule. You can also see that capsular space that gets filled with filtrate as it filters through the glomerulus. You can also see some of the granular cells in the walls of the afferent arteriole, as well as some of the macula densa, kind of salt sensor cells. Both of those things are part of the juxtaglomerular complex. One thing to note about the renal tubules, it's what you see in cross-section kind of surrounding the renal corpuscle. If the lumen of the tubule looks kind of fuzzy, like these two here, those are likely to be cross-sections of the proximal convoluted tubule. The proximal convoluted tubule has lots of little microvilli, little finger-like projections that create more surface area for reabsorption, and so the lumen kind of appears much fuzzier. Whereas if you have a lumen that's much clearer looking, then you're likely looking at either part of the nephron loop or the proximal convoluted tubule, because they don't have as many microvilli. The next organ, in terms of looking at the histology of the urinary system, is the ureter. This is what is going to kind of transport the urine from the kidneys to the bladder. It's kind of a 3-layer wall. So there's, well, really a 4-layer wall, because there's 2 layers in the muscles. So there's a mucosal layer, which consists of transitional epithelium and then a little underlying connective tissue. And then there's 2 layers of muscle, a circular and longitudinal layer. So one layer that kind of wraps around the ureter in cross-section, that's the circular layer, and then a layer of muscle that goes along the length of the ureter. And then wrapping around all of that is some connective tissue adventitia to kind of just protect the ureter and help anchor it in place. If we look at the bladder, that's what comes next. You'll see a very similar kind of setup to what we've seen with other organs. So there's a mucosal layer of transitional epithelia, again with some little underlying connective tissue in the lamina propria, and then actually 3 layers of muscle. So there's an inner longitudinal layer that goes the length of the bladder, there's a circular layer that wraps around the bladder, and then there's another longitudinal layer kind of going with the length of the bladder again. The wall of the bladder is actually very, very muscular. You'll also see that if we look at the mucosa, it's not really smooth, it's kind of folded. So if you were to open up the bladder, you would see lots of what we call rugae or folds, which help the urine store, or help the bladder store more urine than it otherwise would. And although you cannot see it here in this particular slide, just as there is an adventitia layer around the ureter, there is an adventitia around the bladder as well, kind of some connective tissue for protection. One of the things that you do want to be able to do, in addition to recognizing the layers of the wall of the bladder, the tunics, for instance, if we were talking about blood vessels, is to differentiate between a bladder that is collapsed, contracted, empty, and a bladder that is distended or full. One thing, the rugae, the folds in the walls of the bladder, will flatten out in the full bladder, whereas they will be present and visible in the empty bladder. But you also can look at the transitional epithelium itself. So in an empty bladder, the big, floofy, kind of squishy umbrella cells are all squishy looking, right? They're kind of big and rounded, and there's like a dome shape to them. Whereas in the distended bladder, the weight of and the pressure of the urine kind of smushes all those umbrella cells, all those epithelial cells down. So in addition to kind of flattening out the walls of the bladder itself altogether, there's a physiological difference, or an anatomical difference, between a bladder that is empty and a bladder that is full.